Hi, my name is Monica, and I'm going to walk you through this poster titled Identifying the Regulatory Networks that Model the Transcriptional Responses to SARS-CoV-2 Infection in Humans. As an introduction, the pathophysiology underlying COVID 19 hasn't been fully understood. Although key genes and pathways have been described, we have not yet come to a consensus over how the cellular machinery assembles into gene regulatory networks to counteract the infection. Notably, it has been less described across tissues. Briefly, here we integrate RNA-seq transcriptomic data from different studies regarding SARS-CoV-2 infection to build regulons, disease transcription factors with their target genes, and search for differential activation. We selected four bulk RNA-seq datasets: one, exploring viral infections across lung epithelial cell lines, and the others with COVID 19 or healthy tissue data. When possible, data was processed through the same pre-processing pipeline, or this pipeline was adjusted to share methods with data. The data was split into three different analyses: one for cell lines, another for lung, and another one for all tissues analysis, where combat-sec was used to correct for batch effects. We then used Pisonic 100 times to build robust regulons, test for differential activation, and characterize them. Here, we see the overall transcriptomic profile of each analysis in PCAs. Panel A shows the cell lines data. We see that the first component separates experiments by low, green, or high, pink interferon response, shown by the location of silencing or overexpression experiments of interferon, with SARS-CoV-2 infection samples characterized by the latter. Panel B shows tissue data. Blue are healthy samples and red are COVID samples. The first component reflects healthy infected states, but also a technical bias in source datasets. Samples are also grouped by source tissue. Panel C shows lung samples across datasets. COVID samples in red are very heterogeneous but are mixed across studies. Healthy samples in blue grouped together. After building the regulons with Xenic, we see here the cell lines analysis. In panel A, we see an offset plot of the regulons that are distinctly shared across infection or treatment experiments, each of which show a unique regulatory signature. Panel C is a heat map of the regulons enrichment per experiment in Y, and in the x-axis, the TF guiding each of the enriched and upregulated regulons. We see a particular signature in SARS-CoV-2 infection experiments, particularly RX, RG, and SMARCA5 regulons marked in green were the only two regulons that are uniquely shared across three SARS-CoV-2 infection experiments, and their gene ontology biological process enriched terms, shown in B, mention expected activities, such as viral defense, interferon silencing, cell death, and differentiation. For the tissues analysis, panel A shows again the enrichment of regulons across COVID 19 tissues, and in the x axis, the upregulated and enriched regulons in heart, red, liver, green, bowel in blue, or kidney in purple. As expected, we see that almost all regulons present in these tissues are also active in lung, so only KLF14 regulon is uniquely activated in liver and no other tissue during infection. In the lower bar, highlighted in pink, a set of 22 of these regulons are shared across lung, heart, and liver COVID samples, also shown in offset plot in B panel. And per regulon enriched GO terms in panel C indicate an increased immune and growth response. In the lung COVID 19 analysis, we found a short set of upregulated regulons taking data from three independent sources. In brighter pink are those that are also found in the previous tissues long analysis, and in B are shown their enriched biological terms related to a high innate and adaptive immune response. As conclusions, we saw that SARS-CoV-2 infection is characterized by a high interferon response, as we knew. We also identified through SARS-CoV-2 uniquely activated regulons guided by SMARCA5 and RXRG transcription factors. We also saw a strong activation of regulons in liver and heart, 22 of which are shared with lung. Co-enrichment analysis suggested this drive a tissue repair response. At last, COVID-19 lung regulons constructed with data across studies include non-inflammatory genes and elucidate novel guide transcription factors such as DRGX and CNF467. At last, I'd like to thank you for your attention, and I'll be happy to take any further questions.